Hi, I'm Jill Quigley. I'm the Chief Operating Officer at PASG Bio. It's really exciting to be here with a group of people, the first one that I've been to since we shut down for COVID. I joined PASG Bio in 2018 as the first employee. I came with the founders, Steve Squinto and Jim Wilson and Tashi Yamada. And it's always exciting to be able to come and talk about what we're doing and where we've moved to from those early days in 2018. So with that, let's start with our legal disclaimer. I will be making forward-looking statements during this presentation. So PASG Bio, who are we? We're a company that's focused on patients, and we have been since the very beginning. Our vision is to fulfill the promise of gene therapy for patients with devastating CNS diseases where there's significant unmet need and not great treatment options or no treatment options. We have programs in both infant and adult disorders, and we have programs in rare and large diseases. Underlying the company is a corporate model that we feel very strongly sets us up for success. And that starts with a deep relationship with the gene therapy program at the University of Pennsylvania, which of course is led by our co-founder and chief scientific advisor, Dr. Jim Wilson. He and his team of around 300 people now are deeply partnered with us as we look at advancing technologies, next generation technology, capsid designs, and look to apply this knowledge and development translational science, experience to CNS disorders that affect patients with rare and large disorders. This ties directly to our pipeline. We have 17 options to exercise with the gene therapy program. To date, we've exercised 7. So we have 3 programs that are in clinical stage and 4 that are in research stage. We also have exploratory research programs in Alzheimer's disease and temporal lobe epilepsy. That leaves us with 10 remaining options with Penn to exercise, and we can apply new technology to these new programs. We can also apply the technology to our existing programs. So we have a very flexible pipeline, which excites us and allows us to deliver for patients. From the very beginning of the company, we've been focused on manufacturing. We can have a solid science foundation, but we also need to be able to deliver in manufacturing. We can't be successful without that. And so we've worked from the very beginning of the launch of the company to have a flexible and scalable manufacturing control. We started exercising our strategy by adding a dedicated GMP suite at Catalent. And most recently this year, we've added an analytical lab with state-of-the-art technology to support both CMC and our clinical programs in Hopewell, New Jersey, called Princeton West. The company is very well positioned financially. We have robust capital, but importantly, we've been able to recruit great people. Can't deliver on science, can't deliver on manufacturing without the people. So we've been able to recruit leaders in neurodegenerative development and also people who have very deep experience in gene therapy manufacturing. And we're very proud of the team that we've assembled. Digging a little bit deeper just for a minute into what we feel differentiates us when we think about our path for clinical success. We think about our pipeline as having a higher probability of technical and regulatory success. And the reason for this is really our ability to have data-driven decision-making and our relationship with Penn and the people that we've recruited. So when we think about the gene therapy program, Jim Wilson has decades of experience, of course, in gene therapy. He has an A+ team. They're able to help have insights into R&D, optimal capsid, transgene promoter. We think about the patient need. We tailor the product, the therapy that we're going to use in a program based on what we find in preclinical safety and efficacy data. Our candidate selection process is very rigorous. And this work that is going on with Penn and the way that we're selecting our programs and the capsids and the therapeutic we're going to use and the delivery option we're going to use all underpins each of our programs in our pipeline. So our lead 3 programs all use ICM delivery. That's an injection at the base of the neck, which goes right into the CSF. This allows us to avoid crossing the blood-brain barrier and some of the other toxicities we've seen in other programs. We've divided here the pipeline into pediatric and adult indications. Our lead, most advanced pediatric programs are in GM1 gangliosidosis and Craba disease. We were really excited in April of this year to announce the dosing of our first patient in our GM1 study. And we are set and on track to deliver initial safety and 30-day biomarker data for that first cohort in GM1 at the end of this year. For Craba disease, we've opened clinical sites, and we're in the process of screening, and we do expect to enroll a patient before the end of the year. We expect to have data in that program, again, initial safety and 30-day biomarker data in the first half of 2022. MLD is in preclinical phase, and CMT2A is still in discovery. Turning to the adult programs, FTD is our most advanced program in our adult pipeline. We have clinical sites open for our FTD granulin program. We do expect to enroll a patient in that study by the end of the year and have initial safety and 30-day biomarker data by the first half of 2022. ALS and an unnamed CNS indication, which you can think of as being similar in size to FTD, are both in discovery. And then we have target ID ongoing for programs for Alzheimer's disease and temporal lobe epilepsy. Again, this leaves us with 10 options to exercise as we think about continuing to build the pipeline. And because of our relationship with the gene therapy program and the support of their discovery work, we're able to apply new technologies to our existing programs if we feel they will be improved down the road by those, and to new programs. And so it gives us a lot of flexibility in terms of how we can deliver the best product for a patient. I'm just going to give you a high-level overview of the 3 lead programs in terms of disease state and clinical trial design. There's not enough time to go through all the data in the time that we have allotted. So for GM1 gangliosidosis, this is, I'm going to use the word devastating repetitively. I need to find a new word, but it's a devastating disease. These children have a neurological lysosomal storage disorder. It's caused by GLB1 mutations, and it's characterized by destruction of neurons in the brain and the spinal cord. This is rapidly progressing. It's very sad. They wind up with reduced muscle tone. They have progressive CNS dysfunction, deafness, blindness. It's a rare disease. It's about 1 in 100,000 live births, and there's no disease-modifying therapies approved for this today. In terms of the clinical study design, again, this is using a single dose delivery through ICM. It's a phase 1/2 open label study. It's a dose escalating study. You can see the cohorts and how they lay out on the slide that's being presented. Our primary endpoints here are safety and tolerability and efficacy. And again, expecting to read out our first cohort by the end of this year. I'm looking forward to that. For Craba disease, again, this is another rapidly progressing, very sad pediatric lysosomal storage disease. This is caused by GALC mutations, and it's characterized by demyelination in the neurons of the brain and in the periphery. It's the same thing. Disease progression here is very rapid. These kids stop gaining milestones. They have staring episodes, deafness, blindness. This is also a rare disease, and there's no disease-modifying therapies that are presently approved. We expect to dose our first patient by the end of the year. Clinical study design here will look similar to our study design for GM1. Again, looking at 2 different age ranges, phase 1/2 open label, ICM delivery, primary endpoints being safety and tolerability and efficacy. For FTD, our first adult neurodegenerative disease. This is a slightly larger rare disease, so it's about 5 to 10% of all FTD, which is about 3,000 to 6,000 patients for prevalence. It's caused by the GRN gene mutation, what we're focused on, and this leads to a progranulin deficiency. This disease, all the diseases that they're all rapidly progressing, and here, these adults, they lose speech, they lose executive functioning, and they have very severe behavioral changes. A lot of times, this is misdiagnosed because of that. Uplifted, our clinical study here, we expect to enroll our first patient by the end of the year. Again, dose escalation, phase 1/2, open label study, reading out on safety and tolerability and efficacy, also a single dose ICM delivery. So when we think about what's coming up ahead, I've mentioned this, so it's the same thing I've already said. If you've been paying attention, you could do it for me, which is we've dosed our first patient in GM1. We announced that in April of this year. We expect to read out the data in that first cohort by the end of the year. We expect to dose in Craba and FTD by the end of this year and read out the data in the first cohort by the first half of next year. We are continuing to advance the other programs in our pipeline, so MLD, ALS, CMT2A, and our other undisclosed CNS disorder in adults. We're continuing to advance our target ID programs for Alzheimer's disease and TLE. And we continue to evaluate and look to build our pipeline. We want to be able to reach and help patients, and so we continue to look to do that. And I thank you for your time, and I'm happy to take any questions in the few minutes we have left.